So again, thank you so much for a warm welcome on a cool day. And what I'd like to do today is just talk a little bit about Gen 2 renewable fuels. Gen 2 being cellulosic derived, and renewable, of course, speaks for itself. I'd like to acknowledge, first of all, that the work that you'll see, it's a high-level overview of some of the work that we have done together with my collaborators, has been supported by the Department of Energy, by Purdue University, by the USDA, also the College of Agriculture and Engineering at Purdue, and Moscoma Corporation. So this is a short quote from the Wall Street Journal, October 2nd. What's really surprising is that the U.S. now, would have been surprising a couple years ago anyhow, the U.S. now will be overtaking Russia as a major oil supplier. And in fact, the shale rock formations of oil and gas that are now being basically developed have a huge potential for this country, if it wanted to actually become energy self-sufficient for a short period of time. So things have really changed. The reason I point out is often you'll have heard that renewable fuels are necessary for energy security. So at least temporarily, this particular argument may not be as strong as it might have been 5 or 10 years ago. And of course, the reason for this increase is what we call hydraulic fracturing, and this has been actually developed over a period of almost 25 or 30 years. And so suddenly it's on the scene, but in fact it's been developed, and these fields have been developed over a long period of time. Now they're becoming very productive. And I think if you look at renewable fuels, you'll probably see a similar history. Now, something else that may be surprising to some is that US CO2 emissions are actually decreasing. And again, this is a curve from the EI, the Energy Information Administration, DOE's website. And what you can see is starting in 1990, the CO2 emissions, annual percent change increase was 15%. And the CO2, to be clear, is increasing, but the rate of change now is again decreasing. And so as a consequence, the argument that we need renewable fuels to decrease the rate of CO2 emissions may not be as strong as it might have been. Now, on the other hand, this is very interesting. This is called a Keeling Plot. And what it shows is the total CO2 globally. And what you can see from 1960 until 2010, it's increased to the point where it is now close to 400 parts per million, which is very large. And so worldwide, the CO2 emissions are increasing. So what we're seeing in the US is the effects of natural gas use. Natural gas generates less CO2 per unit energy. Also, unfortunately, due to the slower economy over the last couple years, we have less energy use. But what's really interesting, if you dig into the DOE report, the energy intensity of certain products is decreasing. In other words, these products are being made with less energy input. Yet worldwide, CO2 is still increasing, and so as a consequence, we have to be, in fact, very cognizant of this, because the CO2 levels and climate change that they may cause in fact is global. So the energy consumption in other countries is also increasing. And here you have the United States, India, and China, and the US energy consumption is projected to be fairly flat. But the energy consumption, this is all forms of energy, by the way, including electrical energy, will be increasing. Now, if you look at petroleum and other liquid fuels again, the developed countries, which is a dark line, are fairly flat. The developing nations, however, not surprisingly, as their standard of living goes up, the technology improves, and manufacturing changes, they will be increasing their energy consumption of liquid fuels, mainly for transportation purposes. And so as a consequence, what's happening in the US is quite a bit different from what's happening in the rest of the world. So basically, last point here is that the world energy consumption is increasing for all types of energy, and you can see nuclear, renewables, natural gas, coal, and liquid. And so the reason that we should be very concerned and working hard to find renewable, sustainable sources of renewable energy, particularly for liquid fuels, is the fact that the overall world consumption of energy will continue to increase. The overall world generation of CO2 will also continue to increase. And that, together with other types of emissions into the air, may or will cause climate change, which has a time constant which is much shorter than we may be able to react to it. So again, all these factors give you a lot of reasons why we want to continue working in this. Now, the other interesting parameter to look at is price. And so if you want to develop sustainable energy sources, one needs to build plants that manufacture these particular liquid fuels and manufacture or generate the energy. But to do this, one needs investment. And investment really, particularly for something that's technologically risky initially, investment also would require some sort of certainty on pricing and market demand. And this is again from the DOE website, and you can see that if under certain scenarios that we could have a low oil price, a reference oil price, and a high oil price. And these oil prices change from $75 a barrel to close to $250. Now, if somebody was looking at this and trying to make an investment in renewable energy, they might say, please come back later when we can, in fact, model what the impact might be and what the possibility is of you repaying the loan, if that's what you're doing, or the equity, somehow having a return on the equity for building a plant. So this means that we have to do a very good job in eliminating all of the other uncertainty, and the part that we work on, I work on, all of us will work on, is the technology part. But we have to be cognizant there's a lot of uncertainty in this market, and that's why there's so much debate of what should we do as the next step. So what is Gen 2 second generation biofuels, and why are there such differences or major uncertainties? Well, there's future oil prices. There's feedstock costs. This is of the feedstocks that would be used for cellulosic biofuels, and availability by region, although this is now becoming a bit more narrow. Conversion costs and efficiencies. This depends on the type of process one would use. A bioprocess could be used, a chemical, thermochem, and so forth. Environmental impacts, and there's a lot of debate on that as well. Some studies suggest that renewables will have a negative environmental impact. Some studies say it will be very, very positive. Actually, it really depends on the type of technology, what it's replacing, and where the renewable energy is sourced. Then there is government policy. And again, I think you read the newspapers, and there's a lot of debates of what to do with the RFS. That's the Renewable Fuel Standard, which basically mandates the use of renewable fuels, particularly ethanol. And also, just in general, what the environmental policy will be long-term. And again, these introduce more uncertainties. And so this is why you have to admire the companies that have been able to build the first cellulosic plants and get the investment, and the Department of Energy, which has supported some of these programs. So the combination of all of these uncertainties, and I've named a few, makes the analysis of the long-term impact of biofuels just about as uncertain. There's a big beta. And what we have to do is try to bring this closer so that we, in fact, can encourage investment. And this has a lot to do with research, technology, and doing good fundamental science. But the current condition of the financial markets also causes difficult conditions for those of us that would like to buy a house or get a mortgage. And that's very, very low risk. It's still difficult, if you read again some of the newspapers. And so if the banks are really reticent on loans that historically have paid back well, and you have something tangible for it, it is easy to understand why right now we're in a little bit of a lull, but I think these things will change as the economy picks up and some of these other areas are better to define. So what is Gen 1? Gen 1 is ethanol from corn, from wheat, from the sugar that's extracted from the sugar cane. And there's approximately 28 billion gallons, I put greater than 25 billion gallons in the last year, that are produced and consumed, mainly for fuel purposes. There are some beverage grade products included in this total because of the way the surveys were done, but the majority of the fuels would be used for liquid fuels. Now what's interesting is sugarcane ethanol. This is where you take the large sugarcane bagasse. It looks like a corn stalk with no corn. It's cut down, it's ground up, and then they squeeze the sugar out of it. That sugar then can either be made into sugar, refined into sugar, depending on the prices, or fermented into ethanol. And most of it is, that I'm familiar with, is produced in Brazil. So it turns out, even if they ferment the sugar itself and the way it's grown and processed results in a 61% reduction in greenhouse gas using the current life cycle analyses. And I was surprised at this number, but I went to the Unica website. So this is why Brazilian ethanol can in fact be imported into the imported by the US, and be considered an advanced biofuel, and therefore qualify, at least in theory, could qualify for it doesn't, but qualify for subsidies for advanced or cellulosic biofuels. And the reason is there's very little processing required to extract the sugar out and make it into ethanol, unlike corn or cellulose where you have to do cooking, you have to do grinding, you have to transport it, and so forth. Okay, so that may be a little bit surprise. Now Gen 2 biofuels, which is what we'll talk about today, are cellulosic, and they're derived from renewable resources that are not used for food for the most part. Things like corn stalks, sugarcane bagasse, wood, purposely grown and developed energy crops like switchgrass. And right now, the production by the end of this year, instantaneous production, will be about in the range of 10 to 20 million gallons per year. Now, you might laugh at this number, 25 billion, 10 million. Okay. The reason I point this out is this reminds me of what happened in fracking. At first, they had very small fields. They had a few experiments up in the northern US. And it took a long gestation period. And suddenly, we have this huge amount of fuel. Similarly here, what's really significant is the first plants that are making cellulosic ethanol are running today. They're selling it commercially, and they're starting to get very valuable data. And this is as a consequence of the program by the US Department of Energy, EERE, or at least that's what they used to be called, they're now called BETO, I believe, group, okay? So this is very important because this will give the information to start reducing the risk on offtake, feedstock, processing, and so therefore it also reduces the financial risk. So we might go back about 3 years, and some of us in this room actually participated in this study. I worked on the part that had to do with bioprocessing and helped write the chapter together with Greg Stephanopoulos, Jim Katzer, who unfortunately recently passed away. And we looked at the bioprocessing part, but I thought you might be interested to see what the national academies thought about the situation back in 2009, which is just a few years ago. And these were experts from the oil industry, agribusiness, chemical engineers, mechanical engineers, and so forth. And their big concerns, our concerns in 2009 were environmental, national security, and economic competitiveness. Now, if you read the bylines, the context has changed, but still our concern today should be environmental, national security, and economic competitiveness. And I won't go into the details, but so the rationale behind all this still remains, although the context, as I say, has changed. Now, the estimated US lignocellulosic feedstock that could be potentially produced for biofuels in a sustainable basis, this is from the National Academy study, is roughly about 500 million tons a year, which is actually enough to make 30% of our liquid fuel consumption. And this is sustainably harvested, it's available, we did all sorts of surveys on that. And then if you put in the animal manure and municipal solid waste, it's a little bit higher. And in fact, at the time I said, why would we be able to utilize municipal solid waste? It turns out the first cellulosic ethanol plant that's running in Florida, Vero Beach, being run by INEOS, uses municipal waste, grass clippings in this case, is sited next to a municipal waste dump. And the way they do it is they gasify the material, and then they ferment the gas, CO and hydrogen, into ethanol. And it's running today. So here's a large source, and this is kind of nice because there's no input cost and probably a negative cost because people will pay you to take it. Whereas the other feedstocks, you have to pay the producer. So how do we harvest it? Are there systems answers? Yes. This is some machines that harvest corn and stalks at the same time. These were developed with Poe at ADM, John Deere, I believe, but anyhow, there's been a number of studies done over the last 7 years. And this is, by the way, purposely grown, I think this is switchgrass. But anyhow, the yield here, if it were grown for purposes of fuel, would be between 5 to 7 tons per acre. And as a rough metric, you can get 100 gallons of ethanol from a ton of biomass. So this is quite a bit of biomass. And last but not least, this is from Purdue. And long story short, we're growing hybrid poplar. We've talked about this to some people anyhow today. And these poplars will grow 6 feet a year. I realize I'm on web, so I hope my wife isn't watching this. But I had proposed to put these trees in our front yard because we'd have a 20-foot tree in about 3 years, and she nixed the idea, okay? Because what these plants will do is actually, if you cut them down, they'll sprout. They're called a coppice plant. And these are already being used widely for making certain types of fiber, for tissue paper, et cetera. And in fact, they grow very quickly, they have minimal fertilization, and they're an excellent feedstock, as it turns out, for making ethanol. And again, these are not genetically modified, all right? So there's test plots being tried for that. So the question then is, if you look at the feedstock compositions between poplar, red maple, that's hardwood, cornstover, and switchgrass, if you look at the bottom line, the yields are roughly the same. This assumes 100% conversion, 100% metabolic efficiency in the fermentation. That won't occur, but this gives you a metric, a measure, okay? So roughly you're looking at between 0.35 and 0.4 tons of ethanol for each ton of dry biomass. So the efficiency with which the carbon is converted is actually very good. And this number actually corresponds to, we'll call it American Standard Units, and that would correspond to roughly 100 gallons of ethanol neat basis per dry short ton of biomass material. And the key components that are converted are cellulose, and you can see why we like wood, because it has a much higher glucan cellulose content. It's harder to get out at it. And then also the other ones are xylan, arabinan, acetyl, and these are all part of what we call hemicellulose. So roughly 60% of the material is carbohydrate, 40% is lignin, extractives, ash, and other things. So again, if you look at the yield of 0.4 tons of ethanol per ton of dry biomass, the efficiency of the carbon that's converted is very high. But the question then comes up, how much material do we need for a plant? And are there places in the US that could sustainably, for 25, 30, 40 years, provide the feedstock required so that the plant will run day in and day out? And so the other thing that the National Academy study carried out was a survey to find out how many sites would be able to, on the left, provide 800 to 2,000 tons a day, 2,000 to 4,000, so forth, up to 7,000 tons a day, which would be a very large plant, probably the largest feasible. And so it turns out the siting of the plants is limited to somewhere between 17 and 162 places in the US. And the reason we're doing this is you have to have a certain economy of scale once these first plants prove the technology that will in fact give the types of returns that will enable the financing to be carried out. So this basically addresses the question, you can only ship biomass a short distance, between 50 to no more than 100 miles. And so you have to have a radius where not only can you get the feedstock this year, you have to have the feedstock available indefinitely. So that means only part of that acreage would be utilized or harvested, and there's certain plant management, forest management or field management practices that would have to be instituted. Okay, so there's a limited number of places in the US where you can build a plant that is large enough to make economic sense from which you can get the feedstock sustainably. But still, this is quite an impressive number, right? And again, 1,000 tons of biomass, even with reduced efficiency, is equal to 200 or 300 tons of ethanol. So what does a classical, whatever you want to call it, classical cellulose to ethanol process look like? Starting on your left, there's pretreatment. What does pretreatment do? Imagine you have a big log, you run it through this grinder, and it comes out with small chips. That's the first step. Then what happens is you take these chips and you mix, in our case, the technology I worked on at Purdue and with Moscone. In our case, we use water, no chemicals added, cook the wood chips basically in this water under pressure, and then basically then take the solid material which is obtained, and that then goes into the next step. And depending on the technology, the hydrolysis can be carried out, and then fermentation, or they can be done at the same time. What the hydrolysis does is it takes the carbohydrate, they're called structural carbohydrates, and converts them into monosaccharides, glucose and xylose principally. These are then fermented to ethanol, and the fermentation is made possible by genetically modified yeast that have introduced into them a pathway that utilizes not only 6 carbon sugars, which a native yeast will do, but also 5 carbon sugars, which native yeast generally will not do. And so there are a number of yeasts that are available now that are very efficient in both converting both glucose and xylose to ethanol. After that, the rest of the process looks very much like a current corn ethanol process. You have separations. A distillation is carried out. It's followed by a use of a molecular sieve to remove the last trace of water. And then the solids that remain are lignin. They're very energy dense, and they can be used for combustion or gasification to generate electricity and provide energy for the plant. In fact, the amount of lignin that is left over, given a fairly efficient process, is sufficient that you actually can export heat or electricity from this plant. So part of the economics of a process like this is what you do with the co-products. So that's your basic process. So 4 steps in the bioprocessing. 5th step is pretty well-proven separation. 6th step is the utilization of the lignin. And basically what is a modified wood or coal-burning electrical utility. So how do you combine hydrolysis and fermentation? There's 2 ways to do it. You take the enzyme and the yeast, you combine the 2, and then carry out hydrolysis and fermentation at the same time. The reason for this is that the glucose itself, or the cellobios, those are the monomer and dimer that's derived from cellulose, are very inhibitory of the cellulase enzymes, the enzymes that hydrolyze these carbohydrates into fermentable substrates. So the idea is if you ferment them away to ethanol, you reduce the inhibition, and you end up using less enzyme. The other way to do it, however, is to genetically modify yeast, introduce the genes for making the cellulase enzymes, and then just having a single yeast that can ferment the sugars, but also generate the enzymes required to make the sugars from the cellulose. And that was developed and espoused by Lee Lind. And Lee Lind, Charlie Weinman, and Bob Johnson were the founders of Moscone. And Lee is, I think, and as is Charlie, I'm sure, very well known to our audience here. And so basically it combines a simple pretreatment with bioprocessing and distillation. So really what consolidated bioprocessing is the direct microbial conversion of cellulose to ethanol. This technology is currently being used in the corn industry in a technology called MGT, where yeast is added that, in fact, makes the enzyme required to break starch down into ethanol. But this is very important because it proves that the yeast can be made, and it proves the technology is working at a large scale, although it's for a different application. So the issue, though, the challenge we still face is enzyme costs need to be lower. And so there's various ways to get at that. We reduce the amount of enzyme used. That's called loading. We do protein engineering in order to improve the specific activity. the number of times that an enzyme per second, the number of times that an enzyme can move along in hydrolyzed glycosidic linkage to give us soluble sugars. We can increase the yield, and this would be mainly in the ethanol fermentation part, but again, if you have a certain amount of enzyme, you get a certain amount of ethanol. If the yield of ethanol increases, then the amount of loading per unit production is less. However, yield really, most technologies are bumping up against the theoretical maximum. So there's not a lot of maneuvering room there. There's the cost of production, and that is facility dependent. In other words, it's how you design the facility, how much capital, how much project capital is required for these plants. And then there's also the cost of consumables and labor and raw materials principally being the biomass, which is probably going to be, in a mature plant, 60 or 70% of the total operating costs. So feedstock costs and assuring you have a supply is going to be critical to the economic success of such plants. Now, there are models for calculating enzyme costs, and actually an excellent one was developed by Professor Harvey Blanch at Berkeley. Although published industrial data is not available, I thought that Harvey's analysis was really useful. And so the base case he presented was 20% pretreated corn stover solids, a yield of 70% in 5 days. Now, these fermentations right now take a long time. The corn ethanol fermentation is roughly 2 days. Most of these cellulose fermentations will be 5 days, but the times are now starting to get shorter. Okay, so this is and then 2 cases. Case 1 is 5 FPU per gram cellulose. That would be 10 milligrams of protein per gram cellulose. So these are measures of how much catalyst, how much enzyme we're using. And the second case he talked about was 10, double it, about 20 milligrams per gram cellulose. By the way, both of these numbers are considered to be fairly high. And the reason is, you'll see it from Harvey's analysis, and that is a lot of information on this plot. The type of protein, if we had a protein that had the same cost as soy protein, we have case 1. So we're saying, okay, soy protein is about $1.25 a kilo. And if that soy protein had the ability to hydrolyze cellulose, here's what the cost would be for different yields. Second case is cellulose for 5 FPU or 10 milligrams per gram of cellulose being hydrolyzed. And the third one is 10 and 20. Okay? So if you look at the top line, if the yield that we're getting is 52 gallons per ton, the soy protein would cost 18 cents per gallon. If we got the theoretical maximum yield, 100% everything, which is not going to happen, the lowest cost you could have is 8 cents a gallon. This is if you had soy protein. However, the protein cost that they assumed for enzymes was about 10 times higher, about $10 a kilo, in which case, depending on yield, the cost could range from 73 cents to 34 cents. And if you had to use double the amount of enzyme, and by the way, these are the numbers that I often see in the literature, and if I review the papers, I question the numbers when they say this is low enzyme loading. This is actually quite high. In the best yield case, it would be somewhere between 70 and 80 cents a gallon. That's not going to work long term. And so this is why there's so much emphasis on reducing the cost of producing the enzyme, increasing their activity so you use less of them, and then also making enzyme within the fermentation. And with all these numbers, I think the cost will gravitate to a range that is, in fact, tenable. It's probably going to be in the 20 to 30 cents per gallon range, although it's hard to predict. So there's a lot of research to be done to improve this. Now, one way to get more money, more bang for your buck, if you will, using enzyme hydrolysis is to carry out pretreatment. So this is a cartoon that represents the cellulose structure as a morphology containing cellulose, which is crystalline, that's the black lines. Hemicellulose, which is in fact layered between the cellulose polymer and connected, in some cases, chemically to the lignin. And the purple itself is a lignin itself. So what pretreatment does is it opens up the structure, it dissolves out the hemicellulose, and also disrupts somewhat the structure of cellulose itself. And so by having this opened up, what makes possible is if you add enzymes, which are relatively large molecules, large catalysts, they can access the cellulose in order to cause hydrolysis. So pretreatment means that you get something that's a bit more reactive, or actually a lot more reactive. Pretreatments here in the state of Michigan, for instance, Bruce Dale has a very clever approach using what they call AFEX, ammonia freeze expansion. It's not explosion, tells me expansion. My research for the last 30 years or so has been on using liquid hot water, and each different feedstock has benefits. There's also dilute acid, and there's some others as well. The ionic fluids now being developed under, I think it's in J-Bay, right? Or in one of the DOE centers. So anyhow, there's lots of ways to go, but again, one needs to keep an eye on the cost. But once you get there, you have to realize these little green dots, green line, they represent xyloligosaccharides. Xyloligosaccharides, guess what? Are not cellulose. They bind cellulase very well, but no hydrolysis occurs. It's called inhibition. So what we're starting to look at from this point of view is that the plant itself has many lines of defense against pathogens attacking it. And these lines of defense include the hemicellulose, the acetate, the lignin, the phenolics, the crystalline structure, the morphology of the cells. And what we're trying to do is overcome all of these after we harvest the plant and do it in an economic way. So the rest of the story today will be on some of my research and how we are analyzing what's going on. So as far as what is liquid hot water pretreatment, the conditions are 160 to 220 C. It's subcritical water, okay? Anywhere from 10 minutes to 19 hours residence time in the research that we've done. The severities range, and the severity is a measure of time and temperature pretreatment, range from 3.8 to 4.6. And the pH control itself, we originally added buffer. What we found out that most cellulosic materials, because of their composition of ash, hemicellulose acetate, actually are self-buffering into a pH range that we find attractive, between 4 and 7. Okay, and so what we do is we fill up the tube probably about 80% of the way or so. 37 grams of cellulose, this is a lab result, 37 grams of cellulose, and then we cook it in a sand bath. And then in order to quench the reaction, we put it in a bucket of room temperature water. The temperature drops very quickly, and we get our pretreated material. The whole idea behind this, however, is we want to minimize hydrolysis, so we minimize the formation of monosaccharides, which minimizes aldehyde formation, which are very severe inhibitors of the fermentation by yeast. So if I had to characterize my work, Mark, I guess it's running away from inhibitors, right? So the other question is, can we scale it up? And so we very quickly came to the point where we needed more biomass material, and we had students running 20 or 30 tubes a day. And it was, so what we did is we worked together with a local engineering company, Bowen Engineering. He's a Purdue graduate. And we built a machine in his shop to do the same thing. And basically it had a steam generator, a pipe, a feeder, and then was able to pre-treat cellulosic materials. And this is, I think the capacity here was 3 or 4 kilograms an hour or something like this, but it was enough to get laboratory quantities. Where we ran it was actually on the farm. It was skid mounted. We rented a generator, so it wasn't real energy efficient at that point, and then got the corn stover and actually ran it in a barn at one of the Purdue farms to generate the material. So that also helped us give a useful scale-up data. Well, one of the things we found is we had to have a slurry in order to efficiently hydrolyze and ferment the material. And so this is the material that we started with. And this, in fact, believe it or not, contains 85% water. Another problem with this, this is after pretreatment. It's much darker. Particle size is smaller. And then, without going into details, we used some enzyme to basically liquefy the material so that we could pour it. So that's another, took us a long time to figure out how to do this. It's another detail. You have to have something that can be stirred if you're going to get efficient hydrolysis using catalysts that have a characteristic dimension of probably something on the order of 50 nanometers or so. They're pretty big, right? So basically, one of the reasons you want to do the pretreatment is so that you can get something that is stirrable, pourable, and can be processed further. So what does pretreatment do otherwise for corn stover? Well, if we had no pretreatment, and notice we're using a lot of enzyme here, 15 FPU for these experiments, these early days. Okay, but if we have no pretreatment, this is what we get. If we do the same run and simply cook it, in this case I think it was for 15 minutes, I think it was at 190 C. This is published work, by the way. We were able to get 70% conversion in 24 hours. So again, what you're seeing also is a reduction in time required. So in principle, the pretreatment is required to open up the structure and somehow make the material more reactive with respect to enzyme hydrolysis. Keeping in mind, we still had to use a lot of enzyme at this point. So what about, why are we so concerned about controlling the pH of the pretreatment and avoiding the formation of monosaccharides? Well, the reason is that if we have hemicellulose and cellulose present and we cook it in water, there's several things that will happen. Some of the cellulose and some of the hemicellulose will in fact hydrolyze, because water itself is an acid, by the way, will hydrolyze partially to ligosaccharides. Once we get there at the temperatures we use, they very quickly go to glucose and xylose, and at the temperature we use, they go to degradation products, aldehydes, hydroxymethylfurfural, furfural. The other thing that will happen is that these, the original hemicellulose and cellulose H, C, and C will also form an active form, which we like because it'll hydrolyze easier. But if we don't control the pretreatment reactor, these will move even more quickly into ligosaccharides, glucose, and xylose. And this is why in the first attempts to do pretreatment, in our lab, this is 25 years ago, in others' labs where they used acid, we'd get 50% degradation product. So basically, the other thing that happens is the degradation products themselves contain organic acids, which further catalyze hydrolysis if you don't watch it. So this is why this is called an autocatalytic process. So what the pretreatment attempts to do is to minimize the formation of monosaccharides and thereby minimize formation of inhibitors we don't want and reducing the yields. So in fact, what we have is a reaction sequence that has two stages with increasing severity at each stage and washing between the steps. So the first step, what we try ideally to do is get a physical change and stop it. This is perhaps at 200 degrees C. The second step, we add enzyme and we hydrolyze the active cellulose and some of the cellulose, which is a little bit less reactive, to glucose, but no degradation products. And we have some models for that that have been published and so forth, but that's the principle behind it. So liquid hot water, you control the pH, avoid monosaccharides, and do the hydrolysis at conditions one can control. So the cellulose hydrolysis, however, requires at least 4 different components. The molecular weights of these proteins are between 40 and 60 kilodaltons. There's cellobiohydrolase 1, CBH1. It forms cellobiose from cellulose. And there's two ends to cellulose chain, a reducing end and non-reducing end. The reducing end has the aldehyde linkage. The non-reducing end, the number 4 carbon is sticking out, all right? So CBH1 will attack from one end, CBH2 from the other. So you just imagine they're, right, chipping off C2s, right, cellobiose. And then you have glucohydrolase, an exocomponent that acts randomly, so it forms more of these reactive ends. However, once cellobiose builds up at a concentration of about 100 micromolars, it will inhibit 50% of the enzyme activity. Okay? So what you then need is beta-glucosidase. The beta-glucosidase breaks the cellobiose down into glucose, whose inhibition potential is about half of cellobiose, but still quite significant. The trouble, though, is if you then build up enough glucose, it shuts down the beta-glucosidase. Same problem. So then what you do is you add yeast to ferment the glucose to ethanol, which is basically not very inhibitory of the enzyme. So that's why he tried, so there's several different things going on. Now, what does the enzyme look like, the cellulobiohydrolase? This is a picture from Mike Himmel's group, and he's done some excellent work in actually visualizing how these enzymes settle onto the cellulose and carry out hydrolysis. And so as a consequence, this is a binding domain. This binds to the cellulose. This is a representation of the cellulose structure. This is the catalytic domain. The catalytic domain is where the hydrolysis occurs. So the other problem we have is that cellulose is very, very strong hydrogen bond. So the protein somehow has to get in there and pull it apart and then carry out the hydrolysis. And so that requires a fairly large protein. And we're looking at about 60 kilodaltons here with two domains, a smaller binding domain. The catalytic domain is this big blob. So it turns out that the cellulose chain, which is in red, the active site is in white. So what happens is it pulls the cellulose chain through and then releases a dimer. This is all occurring at 50 degrees centigrade, by the way, which to me is pretty amazing at such a low temperature. And what the active site has, it has two acids, basically. Glutamic acid, one is a nucleophile base, the other is a proton donor acid. So just as a side story, when we saw this part, we actually then developed an organic molecule. It's called maleic acid. We looked at various diacids, which have very similar properties and works well. In fact, gives us very high selectivity of reaction, but at 100 degrees C. So something that this research is leading to is what's called the development of memetics. These are organic molecules that have the same selectivity, are less expensive, in fact can make a very good yield. And again, this is in the early days of this, but so it gives you an idea how this works. So it's a fairly complex picture. But what happens in addition to the pretreatment is we give off other inhibitors. These are phenolics. Lignin is a polyphenolic. And xyloligosaccharides, but we can hydrolyze those away, so reduce the inhibition. But we are then left with phenolics, tannic acid, gallic, cinnamic, ferulic, cumaric, sinapic, vanillin, syringaldehyd, et cetera. These are all products that are formed from the lignin during the pretreatment or released from the cell wall structure when hydrolysis occurs. Now what's really interesting, and we use fairly high activities in order to test this, what's really interesting is if we add tannic acid at the concentrations we see in our pretreated liquid, it in fact will cause the cellulase. This is not the beta-glucosidase, but the CBH and the exo, right? Not the beta-glucosidase, will cause 60% inhibition of the cellulase. And these inhibitors are not metabolized. These are not turned into something else. They're in the background. And what's more interesting is if we get beta-glucosidase from T. reesii, that's one of the fungal organisms that give us a source of the enzyme, the tannic acid basically shuts it down. We're talking about parts per million. So what the tannic acid does is precipitates the protein. So how do you deal with it? You add a lot of enzyme. Well, okay, what you're doing is using enzyme to titrate these phenolics. I mean, it's really interesting. The gallic acid is less inhibitory. The ones that originally put us onto this were publications that ferulic acid interfered with the hydrolysis of corn fiber. And although this was a very measurable inhibition, these others, which are derived from lignin, are even more potent. Now, what's also interesting is not all beta-glucosidases are created equal. So this beta-glucosidase is from A. niger, okay, a different organism. In this case, tannic acid, everything else being equal, only caused about 10% inhibition. So what it says is if we have the right genetic construct, the right protein, we may actually be able to engineer some of this inhibition away and make our enzymes more potent. So there's a lot of study going on that. So why is phenolic so important? Well, let's take a look. Let's go from your right and move left. We form glucose. Okay, we said glucose will inhibit this enzyme, which is beta-glucosidase. Okay, and once this starts to shut down, the cellobiose builds up and inhibits the cellobiohydrolase. And then the cellulose hydrolysis stops. So what we said is we then ferment away the glucose to ethanol, and things work out pretty well. But if we add an inhibitor, phenolics, and it actually deactivates the enzyme very, very quickly, this builds up, hydrolysis stops unless we add a lot of enzyme. So we have to deal with the phenolics. It's a very important thing. But our friends at NREL, Jim McMillan, who's active in AI Chem, had a paper in 2011. So what he did is he washed the material between pretreatment. So at least he removed some of the inhibitors. So what you can see here, and again, this is the amount of protein per gram cellulose. If you had whole slurry, it contained everything, all the inhibitors, the conversion might be about 40. If you wash the same material, wash away the inhibitors, wash away the phenolics, do the same experiment, conversion suddenly is 55. So again, this experiment clearly shows the importance of dealing with inhibitors other than cellobiose and glucose and understanding the chemistry of the cell wall so that we can reduce the amount of enzyme. The reason we're doing this is we want to reduce the amount of enzyme down to the 20 to 30 cent range so that the process is economic. And that's starting to happen, actually. So I like this. This is from a Novo presentation, Novo Nordisk, Novozymes, an enzyme manufacturer. By the way, the beta-glucosidase that they have in the Novozyme 188 is from Aspergillus. So that's why many of us came to the conclusion we should use Spezyme CP, mix in Novozyme 188, and get good hydrolysis, right? Because it turned out the beta-glucosidase resists the phenolics from the lignin, although we didn't realize it at the time until we started getting some of this other data. Well, this is a very interesting plot. Basically, two lines cross, okay, so that means something for sure, right? And basically, the green line is enzyme. So it says if we do a pretreatment using acid going to neutral and then to alkaline, the degree of inhibition of the enzyme will, in fact, increase. Because of the, and we now understand where the compounds are coming from. This is for unwashed material. Whereas if we do dilute acid, neutral, liquid hot water, or alkaline, the degree of inhibition of yeast will decrease. The reason is, as we go to higher pH, we will not form the aldehydes, hydroxymethylfurfural, furfural, formic acid, levulinic acid, which are really strong inhibitors of yeast. The other thing it does is when you're at this end, this will release acetic acid from the hemicellulose, and acetate is a very strong inhibitor of yeast. So as you increase the pH, you're going to release less acetate as well. So the question is, where do you go? And so what our research, our work is focused on is this spot. Now what we want to do is bring this spot up, right? So that's where we were basically looking at pH between 4 and 7 roughly to minimize some of these other effects. Now, what does pretreatment do? And this is sort of an excerpt from lots of data and lots of correlations that we've developed. One thing that pretreatment does that's helpful is it decreases particle size. And this data is a little bit hard to interpret because in addition to decreasing the average particle size, we're also increasing the total number of particles very significantly. But it'll give you an idea. These are different severities, and basically the higher the number, the higher the severity. That means a higher temperature. Time is pretty constant, but let's say it's a higher temperature. So what this shows is that using a large amount of enzyme, again 20 FPU, which is relatively large, as we go to more severe pretreatments, we decrease the particle size and conversion goes up. Makes sense, right? It's more exposed. I should hasten to add, however, we were very careful to wash away all the inhibitors for these runs, which would not be practical necessarily for a process. So the other thing that happens with severity is dP decreases. So again, going backwards. So as the relative cellulose dP is lower, and there's a dP called leveling off of dP, which is about 200 monomer units. Again, we can look at conversions that are in the 80 to 90% range. What's interesting here is Avicel, sulcofloc, and untreated wood. Untreated wood does not hydrolyze well. Sulcofloc, which is not pre-treated by the method we have used here, still gives us fairly good conversion, as does Avicel. So these are outliers, but the reason is these materials do not contain lignin. So they don't correlate very well because we start to lose those other inhibitors that are present. So what sort of picture is emerging? So I thought you're the test audience for this, okay? We'll see whether you like my cartoon or not. So if we start with untreated materials, we have big pores, small pores. Lignin is the dark line. Cellulose, crystalline cellulose with pores are the light lines. These are, by the way, CBH molecules. This is a catalytic domain, right? The black dot is a binding domain. So if we do pretreatments at lower than 200 degrees C, the particles themselves are relatively unchanged. Okay? So we do get some formation of pores. We do get some removal of lignin and hemicellulose. That's the green lines. And we get some access of the enzymes internally into the biomass material. But if we go above 200 degrees C, that's the highest severity data points 8 and 9. What then is happening, and we've seen this physically, is we're breaking up the particles. We have larger pores. The hemicellulose is removed, and the enzyme begins to access the cellulosic material. The conundrum, however, is we're also generating inhibitors, and so we need to control those if we want to reduce the amount of enzyme that is used. So there's a lot of physical effects which transcend any efforts on molecular biology, on the biology of the plant tissue used in the plants that we would hydrolyze, that need to be understood. And I think these are classical mass transfer, kinetics, adsorption phenomena type of chemical engineering problems. And there's a lot of room for improvement here. Because at the end of the day, all these wonderful discoveries need to be translated if they're going to do anybody some good. This is going to require really rigorous chemical engineering scale-up. So the question then would be, why invest now? Well, first of all, we got some sharp chemies at University of Michigan working on it. Okay, that's a good reason. And there's some trends. The major sources of uncertainty, future oil prices. You think oil's going to go, you think gasoline's going to go back to $1 a gallon? Now, I'll give you, I promise, Mark, only one. Okay, when I was young, and I was actually commuting out to Purdue for graduate school, they had gas lines, but the previous year they were selling gasoline at 30 cents a gallon, and some gas stations would give you free glasses if you bought their gasoline. That was in 1971, okay? Now it's what, $3.50? It depends on the day of the week, I suppose. It goes from $3.20 to $4 in our neighborhood. So I think it's a safe bet. We don't know if it's going to be high, medium, or low, but future oil prices are going to go up, right? Feedstock costs, this is biomass costs, are going to go up. Why? Because at some point these plants will start to make feedstock, they will have revenue, and the farmers or the people that manage the forest will say, ah, that's nice, I want a 10% increase. Huh? So the question really is, you have to have a sensitivity of how much feedstock costs will in fact impact the bottom line, the OPEX, the operational cost. Again, sensitivity analysis, risk analysis, classical chemical engineering. Feedstock availability by region. Some regions will have feedstocks that are more easily processed, some less so, some won't be stored as much. And so you have to do an analysis. This is really classical agricultural engineering. But again, and then you have the machinery to harvest it. But the feedstock availability by region, if the price go up, I think the availability will come up. And this is sustainable practice. This is not clear-cutting forests or removing all the corn stover, but sustainable practice. Why am I confident? Look at what's happened to the pulping industry. There is actually a surplus right now in some areas, causing economic issues. Conversion costs, I believe, will go down. I'm excited what I saw here today in the poster session and some of the talks I had with Jeremiah this morning. There's all sorts of cool things combined together that are going to bring the costs down. Conversion efficiency, well, this is saying the same thing. These are not independent parameters. Conversion efficiencies will go up. As we learn more, I think the environmental impact overall of the fuels will go down. By how much, I think people claim we're going to remove all the CO2 from the atmosphere and things will get back to square one. That's probably not feasible. But environmental impacts will decrease. So the question now is government policy. I think long term it will improve. It has to. And that's all I'm going to say. I'm an optimist. And the energy infrastructure takes a long time to develop. And look at the fracking as an example. It's been 25 years, and suddenly, oh, we have a solution here. No, it's been a long time in coming. So what about a summary? Actually, what I'd like to say is that I think low-cost production processes will define cellulose ethanol. If we don't have low-cost production, forget it. All this hard work, the discovery, would be wasted. And I don't think that's going to happen. I think we're going to find ways to translate the technology. And I was really encouraged to hear, just like at Purdue, University of Michigan is looking at the entrepreneurship and startup companies and getting that technology out. And you know what that does? It creates jobs. That creates a good economy. I mean, the bank will load us money to build these plants, right? Then we'll reduce the CO2. So it's really great opportunity for all of us. I think thermochemical processes are defined. Mature technologies, I didn't talk much about that today, but I know that you're doing some research here. I think cleanup is going to be very important. And the catalyst may be expensive, but by combining these two, these may come into a range. There might be hybrids between thermochem and fermentation or vice versa that will come into a range that will be attractive. I think bioprocessing routes right now are more attractive because they use less harsh conditions. They're more selective. They're compatible with a biorefinery concept, that is, make fuel, biochemicals, other products. And low-cost technologies are possible, but not yet proven. So with this, I'd like to end my lecture and just thank everyone for having such a wonderful day here at the University of Michigan. And tomorrow I go to Michigan State. And is Bruce Dale going to be there?
Yes, he will. He's on your schedule for lunch tomorrow.
Okay, yeah. Otherwise, I wasn't going to come. But yeah, so I think we're going to have a wonderful visit at Michigan State as well. And so with this, thank you very much. And thank you to the state of Michigan, I guess.